8M was a patient that contributed to one of the most significant turning points in 20th century brain science. Following a childhood blow to his head, Henry Molaison developed severe seizures. After 18 years, he underwent an experimental lobotomy that removed sections of his medial temporal lobes, including most of his two hippocampi. He no longer experienced seizures, but Molaison was left with permanent amnesia. He could remember scenes from his childhood, but he couldn't form new long-term memories for the rest of his life. After being studied by neuroscientists for over 50 years, he died in 2008 at the age of 82. Despite his struggles, he was a pivotal case study in memory research and led to numerous discoveries in neuroscience. While we don't fully understand how memory and learning works in humans, researchers are learning more and more, in part thanks to HM. Scientists now know that the medial temporal lobe works with other parts of the cerebral cortex to form, organize, consolidate, and retrieve memories. But there are multiple different types of memories, and a different kind of memory are processed by different regions. The hippocampus, perihippocampal region, and areas of the cerebral cortex work together to produce memories of facts and events. Other memories, like emotional or behavioral memories, are handled by parts of the brain like the amygdala, striatum, and cerebellum. All memories can be classified into two different categories, declarative memory and non-declarative memory. Let's talk about declarative memory first. Declarative memory, also known as explicit memory, is a memory for facts, data, and events. Declarative memory can be further divided into long-term and short-term declarative memory. Long-term declarative memory consists of two main memories, semantic and episodic memories. Semantic memories are cultural knowledge, ideas, concepts about the world, and facts. Some examples are names of state capitals, how to add or subtract, word definitions, or historical dates. On the other hand, episodic memory are your own, unique representations of personal experiences, such as recalling the sights, sounds, and emotions of a carnival you went to during your childhood. Interestingly, the emotional significance of your episodic personal memories is mediated by the amygdala and instead of hippocampus. The amygdala is a small structure with two paired almond-shaped regions that modulates the flight or fight responses for survival. Additionally, the perihippocampal region and hippocampus help encode the what of episodic memories. Going back to our case study, HM was able to remember his long-term memories before the lobotomy. However, he was unable to form new long-term memories, but he did keep his short-term memory, which we'll go into next. The second type is short-term declarative memory. Working memory is a temporary type of declarative short-term memory. We use working memory every day. It lets you briefly remember a phone number, a visual image, or some data point needed at the moment or in the very near future. Working memory is partially coordinated by the prefrontal cortex, an important part of the brain. The prefrontal cortex, or PFC, has three main executive functions: attention, decision-making, and long-term planning. Brain imaging studies show that PFC is more active when people concentrate or keep something in their mind, like a phone number. Interestingly, HM did not lose his working memory. He just couldn't create new long-term memories from the working memory. Fascinatingly, the brain's capacity for working memory is limited. It's hypothesized that the max is around 7 pieces of information. Working memory also decays quickly. On the other hand, long-term memory has unlimited capacity. The last type of short-term declarative memory is spatial memory, which is involved in navigational memories and mental maps. Spatial memory is tied to specific types of neurons. Cells called place cells in the hippocampus are activated as you move through a familiar place, like your house or when a mouse navigates a known familiar maze. In fact, studies have shown that mice navigating mazes display specific neuronal activity sequences for left or right turns, which get more distinct as they learn the maze. For unfamiliar new places, so-called grid cells are activated. Grid cells are located in the entorhinal cortex near the hippocampus. They represent coordinates that let the brain track your position in space when you're not familiar with the landmarks or external cues in your area. We talked about declarative memory. Now it's time for the opposite, non-declarative memory. This type of memory is also known as implicit memory. This kind of memory is stored and retrieved without conscious effort. Non-declarative memory includes procedural memory, memory for actions such as when you perform learned motor skills like riding a bike or speaking. You do not consciously retrieve this kind of memory. Motor skill learning, such as procedural memory, involves three main areas of the brain: the basal ganglia, the habit center, the prefrontal cortex, and the cerebellum, which is involved in motor control and coordination. HM did not lose his procedural memory because he could acquire new motor skills, despite not remembering doing them. The brain can create memories and use them to change how it responds to a situation based on experience. We already know that it's called neuroplasticity, but how does the brain rewire itself? Your neurons are constantly sending electrical impulses to the synapses, the small gaps between each neuron where neurotransmitters can trigger excitatory or inhibitory postsynaptic signals by binding to receptors on postsynaptic memory. Encoding new long-term memories involves changes in the number and shapes of synapses, the amount of neurotransmitters released, and the number of receptors on the postsynaptic membrane. These changes are known as synaptic plasticity. The synaptic mechanism for learning memory was first observed in a sea slug, Aplysia californica. It hasn't got many neurons and they're easy to observe. Researchers found chemical and structural changes that correlated with learning and memory, and more studies in the mice found that long-term synaptic structural changes were even correlated to changes in gene expression. The formation of long-term memory employs two genes, the NMDA glutamate receptor and CAMP response element binding protein, or so-called CREB, via two processes: long-term potentiation and long-term depression. Long-term potentiation, or LTP, is a long-lasting increase in synaptic strength that occurs especially in the hippocampus. This is a structural change to the brain that is essential for long-term memory. The exact mechanism depends on the type of neuron, but in general, LTP works by initiating a cascade that increases the number of NMDA receptors on the postsynaptic neuron. On the other hand, LTD, long-term depression, decreases the effectiveness of the synapse. When NMDA binds to glutamate, which you may remember is the most common excitatory neurotransmitter in the brain, calcium ions flood into the postsynaptic cell. These calcium ions function as what's called a second messenger, because their entry into the cell sets off a chain of events. The difference between LTP and LTD is created here. In LTP, more calcium ions flood the cell than in LTD, and this changes the type of enzyme that is activated. In LTP, the higher concentration of calcium ions activate kinase proteins, which can modify the synapse to become more efficient at passing nerve impulses. The increase in calcium ions activates cyclic adenosine monophosphate, or so-called CAMP, which modifies the synapse to increase the number of receptors on the postsynaptic memory. Activating CAMP again and again activates CREB, a trophic factor that modifies gene expression by making changes to the protein synthesis. On the other hand, LTD activates phosphatase enzymes, which make the synapse less efficient at passing nerve impulses. In this way, the brain can encode long-term memories. Interestingly, researchers have actually found that declarative memories are encoded in the hippocampus, then transferred to the frontal lobes for long-term storage and consolidation. Thus, the hippocampus plays a lesser role in retrieving older memories. The frontal cortex assumes the task instead. As researchers gain more insight into the molecular mechanism of memory, we could even discover artificial ways to manipulate synaptic plasticity and memory itself. These methods could be used to treat conditions like PTSD or optimize the way humans learn and memorize things. The possibilities could be limitless. Researchers looked at the brains of prairie voles to study oxytocin, a chemical associated with love. Interestingly, when researchers increased oxytocin receptors, they found that increased oxytocin receptors levels resulted in female voles performing partnerships faster. Emotional memory is another type of non-declarative memory. This is when learned emotional responses become attached to stimuli over time. As a result of repeated exposure, the amygdala, the insula, and the preaqueductal gray in the midbrain are the three most important structures that allow you to feel emotions. Firstly, the amygdala integrates emotions, emotional behavior, and motivation. This allows you to identify reward and punishment, as well as distinguish between friends and foes. Meanwhile, the insula or insular cortex allows you to experience disgust and is also involved in pain. Lastly, the preaqueductal gray is tied with pain perception as well as stress response. The prefrontal cortex, the amygdala, and the insular cortex all project to the preaqueductal gray. Human actions are driven by necessities and rewards. Necessities are things like food, sleep, and avoidance of pain, things your body needs in order to survive. But when you're in a situation weighing risk against reward, like gambling, when you could win it all or lose everything, or in a situation where emotions are running high, the way you make a decision can change. Our responses and actions aren't always logical. Sometimes we make choices under risky or uncertain conditions. That's called effective decision making. And growing area of neuroscience research is investigating how the brain balances reward against risk, and of course, how your emotional state affects this balance. Emotion-centered decision making changes with age, and scientists believe this is because the lateral prefrontal cortex, which is responsible for self-regulation, matures slowly, especially in adolescence. Teens' brains are still developing, and peer pressure is more significant in adolescence than it would be in someone in their 30s or 40s. Older adults might also make more risky decisions because PFC function diminishes with age as well. And of course, now that we've discussed risk, we've got to move on to reward. So let's talk about reward pathways and neurotransmitter governing them. Dopamine is involved in many different processes, including mood, reward, addiction, stress, and memory. The mesolimbic pathway, or the reward pathway, is a major pathway for dopamine connecting the midbrain's ventral tegmental area, VTA, to the nucleus accumbens. Dopamine influences the entire pathway to create emotional associations with rewards. In response to the reward, dopamine signaling may increase or decrease depending on if the reward was higher or lower than expected. Interestingly, researchers found that go-getters, who are more willing to work hard, have a greater dopamine signaling in the striatum and prefrontal cortex, which are both areas known to impact motivation reward. While the brain usually reinforces reward and prevents punishment, aggression can result from inappropriate activation of the brain's reward systems. For example, the lateral habenula responds to punishment by inhibiting dopamine release. Dysfunction of it is linked to inappropriate aggression. Similarly, some regions of the amygdala can trigger rage and aggression. Fascinatingly, when specific portions of the amygdala were removed in lab animals, they became more docile.